Hello everyone. As we have already gone through the digestion and absorption of carbohydrates, proteins, so let's start today digestion and absorption of lipids. What are lipids? You already know very well that lipids they are heterogeneous group of water insoluble organic molecules that can be extracted from tissues by non-polar solvents. They are known as major source of energy for our body and also provide the hydrophobic barriers that permit partitioning of aqueous contents of cells and subcellular structures. They also include some fat soluble vitamins that have regulatory or coenzymes that have important role in the body. They also include prostaglandins and steroid hormones which play a major role in the control of body's homeostasis and other important activities. Lipids also give shape and contour to the body and protect internal organs by giving a cushioning effect and by providing fat. So here is the diagram that shows its composition. We can observe here that more than 90%, almost 95%, they are triglycerides, while other some less proportions are cholesterol, 2%, phospholipids, 5%, free fatty acids, they are 3%, unsaturated fatty acids, dietary fats also included here. Average normal diet for an Asian person that count Pakistan also 20 to 30 gram per day, while Western diet accounts to two or three times of this value. So if we talk about dietary sources of lipids, we can tell that there are two types of sources, animals and vegetable sources. Animal sources, for example, they include dairy products that provide us fat. There is meat, butter, ghee, and similarly fish, pork, eggs that provide us fats. And if we talk about vegetable sources, there are cooking oils such as sunflower oil, mustard oil, groundnut oil. Fats from other vegetable sources can also be obtained. Classification of lipids. Based upon classification of lipid is based upon different factors. So we can say that there are some lipids which are based on reactivity with strong bases. For example, spongifiable lipids and non-spongifiable lipids. If we uh, if we say that there are some lipids which are based on complexity of structure, or if we talk about classification related to complexity of structure, we can say that there are some simple lipids like triglycerides, and there are some complex lipids such as waxes, phospholipids, and there are some derived lipids like steroids, sterols, and in miscellaneous, we can include lipoproteins, glycolipids, sulfolipids, and gangliosides. There is a challenge to the digestion process of digestion and absorption process of the lipids. Lipid digestion and absorption actually it pose some problems, some challenges. Triglycerides, they are large molecules, and unlike carbohydrates and proteins, they are not water soluble. Because of this, they like to cluster together in large droplets when they are in a watery environment like the digestive tract. We know that digestive tract contains a watery environment. So lipids, they like to become clusters in that environment and does not adapt this environment easily. So we can say that the challenges are lipids are not water soluble and triglycerides too large to be absorbed. They are not absorbed directly from the intestinal wall. So the solution of this challenge is that digestive process has to break those large droplets of fat into smaller droplets and then enzymatically digest lipid molecules using enzymes which are called lipases. Lipases, they are specific enzymes used to digest lipids. The mouth and stomach play a small role in this process, but most of the enzymatic digestion of lipids happens in the small intestine. So the solution, the simple solution to this challenge is the process of emulsification. That is to break large droplets of fat into the smaller one. Product of lipid digestion, then they are absorbed easily into the circulation and can be transported around the body, which again requires some special handling also during the process of absorption, since lipids are not water soluble and do not mix with the watery blood. So here the solution is that triglycerides mix with bile and pancreatic secretions. That is, the process of emulsification occurs all here too. The emulsification of lipids is done by bile salts and by the digestive juices and fluids. So let's start digestion and absorption of lipids, how this process occur. What is digestion? You have already a very well idea about it now. Digestion is a process of converting food particles into absorbable form by the help of enzymes present in gastrointestinal tract. And what is the absorption? It is a process of transferring of absorbable food particles or nutrients from intestinal lumen towards blood or the lymphatic system. There are some phases of the digestion and absorption process. The first of all is a preparatory phase, then comes transport phase, and the third one, the last one, is the transportation phase. What is a preparatory phase? We can say that the emulsification, the process of emulsification that we have already talked about, includes in preparatory phase. Actually, this phase includes the digestion of dietary lipids in intestine. Lipid particles are broken down into smaller particles with the help of lipolytic enzymes. Then comes the second one. This phase includes the transport of digested fats across the membrane, the intestinal villi layer that is present into the intestinal epithelial cells. So from this membrane towards the epithelial cells, this is the transport phase. And then comes transportation phase. That includes the events of action that take place inside the intestinal epithelium or its passage through lacteals towards portal blood or towards lymph. That is, say, if we simply understand this process, preparatory phase is the phase in which digestion occurs using enzymes, with the help of enzymes. Transport phase, when these small particles or glycerides, monoglycerides and free fatty acids especially, they transfer from intestinal lumen towards epithelial cells. Then starts transportation phase, where these free fatty acids transfer from epithelial cells towards blood or any circulatory system. Lipids are insoluble in aqueous solution. The digestive enzymes are present in aqueous medium. So this leads certain problems in digestion and absorption of lipids. So we overcome these problems by increasing the surface area of lipids for digestion, by emulsification of lipids with the help of bile salts, and by solubilizing the digested product for absorption. Digestion in mouth. It was believed earlier that very little or almost no fat digestion takes place in the mouth. But recently, in a study, a lipase has been detected, which is known as lingual lipase, as it is present in mouth. So this enzyme is named lingual lipase here. It is important to note that this enzyme, the lipase, it has three names: the lingual lipase, the pancreatic lipase, and the gastric lipase. The lingual lipase, we will recall this enzyme as lingual lipase when it is found in mouth, the gastric lipase when it is present in stomach, and the pancreatic lipase when it is produced in pancreas and secreted in small intestine. So first of all, in mouth, the lingual lipase is secreted by the dorsal surface of the tongue. Hydrolysis of triacylglycerides or triacylglycerols is initiated here in mouth by the action of lingual lipase, which attack the third carbon of the ester bond, forming 1,2-diacylglycerols and free fatty acids, and also help in emulsification. A few things happen in the mouth that start the process of lipid digestion. Chewing, for example, that mechanically breaks food into smaller particles and mix this food with saliva. An enzyme uh, we have already discussed, lingual lipase, is produced by the cells of the tongue and begins enzymatic digestion. So here both the mechanical and the chemical digestion takes place. What is the role of lingual lipase? It is active at low pH, that is starting from 2 to high pH or neutral pH like 7.5. And the optimum pH for its activity, it is 4 to 4.5. And the ideal substrate on which it acts, it is the short chain triacylglycerides. It is important here that milk fat, which contains short chain fatty acids, which are esterified at third position. And this enzymatic action of lipase also continues in the stomach. So this diagram shows the esterification of triacylglycerides with the help of lipase. By adding water molecule, this lipase converts short chain triacylglycerides into free fatty acids and the glycerol backbone. The digestion in stomach. In the stomach, mixing and churning helps to disperse food particles and fat molecule cells in the stomach produce another lipase that is known as gastric lipase here. So as you know that gastric means relating to the stomach. So the name is given to this enzyme with respect to the location where it is produced. It also contributes to enzymatic digestion of triglycerides. The process is the same as we have already seen in the mouth. Lingual lipase swallowed with food and saliva also remains active in the stomach. But together, these two lipases play only a minor role in fat digestion, except in the case of infants. And most enzymatic digestion happens in the small intestine. Lipids are a part of the infant's diet. We know that. Breast milk, for example, it contains about 4% fat that is similar to whole cow's milk fat. Whether breastfed or formula fed, fat provides about half of an infant's calories and it serves an important role in brain development also. Yet infants are born with low levels of the bile and low levels of pancreatic enzyme secretions, which are very essential contributors for towards lipid digestion. So in older children and adult, but what happens in baby when there is very less amount of pancreatic enzyme and bile is secreted? So how the question is how do babies digest all of the fat in their diet? Infants have a few special adaptations to this problem that allow them to digest fat effectively. First of all, they have plenty of lingual and gastric lipases right from birth as compared to the adult one. These enzymes play a much more important role in infants than they do in adults. Second, the second solution is that breast milk actually it contains lipase enzymes that are activated in the baby's small intestine. In other words, the mother makes lipases and sends them in breast milk to help her baby digest the milk fat. Amazing? Is it amazing? Yes. Between increased activity of lingual and gastric lipase and the lipases contained in the breast milk. Apart from this, young infants can efficiently digest fat and reap its nutritional value for growth and brain development. Studies have also shown that fat digestion is more efficient in premature infants, fed breast milk as compared with those fed formula milk. Even pasteurized breast milk as is used when breast milk is donated for feeding babies in the hospital is a little harder to digest because heat denatures the lipases. But infants can still digest pasteurized breast milk and formula milk. They are just less efficient at doing so and absorb less of the products of triacylglyceride digestion. Therefore, fresh mother's milk is the best solution to the diet for the diet of the infants and babies. Now, gastric lipase, what are some specific characteristics of the gastric lipase? It is secreted in small quantities. It is more effective at alkaline pH, that is average pH where at which it is most effective, it is 7.8. The point to be noted that this is the effective pH for gastric lipase and not for lingual lipase. Gastric lipase also requires the presence of calcium for its effectivity and for its action. It is less effective in stomach due to acidic pH, except when intestinal contents are agitated it into the gastric lumen. Not effective for long chain fatty acids, most effective for short and medium chain fatty acids, just like lingual lipases. Milk, egg yolk, and fats containing short chain fatty acids. They are most suitable substrates for its action, such as in children. Gastric hormone, enterogestrone. An enterogestrone is a hormone which is secreted by the mucosa of the duodenum in the lower gastrointestinal tract in response to dietary lipids that inhibits the motion of the contents of chyme. The function of enterogestrone is almost the same as gastric inhibitor peptide. It inhibits gastric secretion and motility of the stomach. Fats delay the rate of emptying of stomach. So action is brought about by the secretion of enterogestrone. Enterogestrone inhibits gastric motility and retards the discharge of bolus of food from the stomach towards the small intestine. So at the end, we have a high satiety value, and this process is very much important during the fasts or during the times when we do not have much to eat. Significance of lingual and gastric lipases. They play important role in lipid digestion in especially neonates, since milk is the main source of energy for neonates and infants. They are important digestive enzymes in pancreatic insufficiency, such as cystic fibrosis or other pancreatic disorder when we do not have enough pancreatic enzymes. So they are the very much important digestive enzymes to digest fats. Lingual and gastric lipases can degrade triacylglycerides with short and medium chain fatty acids in patients with pancreatic disorders, especially, despite a near or complete absence of pancreatic lipase. So, if we summarize this discussion, here we can observe that the enzyme lingual lipase, along with a small amount of phospholipid as an emulsifier, initiates the process of digestion. These actions cause the fats to become more accessible to the digestive enzymes. As a result, the fats become tiny droplets and separated from the watery components. In the stomach, gastric lipase starts to break down triacylglycerides into diacylglycerides and fatty acids, free fatty acids. Within 2 to 4 hours after eating a meal, roughly 30% of the triglycerides are converted to diglycerides and fatty acids. The stomach's churning and contractions help to disperse the fat molecules, while the diglycerides derived in this process act as further emulsifiers. Now comes the small intestine. Small intestine is the major site of fat digestion. This is due to the presence of a powerful lipase in the pancreatic juice. The pancreas secretes pancreatic lipases. This is due to the presence of a powerful lipase in the pancreatic juice and presence of bile salts also, which acts as an effective emulsifying agent for fats. Pancreatic juice and bile enter the upper small intestine, which is called duodenum, by way of pancreatic and bile ducts respectively. The pancreas secretes pancreatic lipases into the small intestine to enzymatically digest triglycerides. Triglycerides are broken down to fatty acids, monoglycerides, and some free glycerol, cholesterol, and fat-soluble vitamins do not need to be enzymatically digested. As the stomach content enters the small intestine, most of the dietary lipids are undigested and clustered in large droplets. Now, bile comes here, which is made in the liver and stored in the gallbladder. It is released into the duodenum, the first section of the small intestine. Bile salts have both a hydrophobic and a hydrophilic site, so they are attracted to both fats and water. This makes them effective emulsifiers, meaning that they break large fat globules into smaller droplets. Emulsification makes lipids more accessible to digestive enzymes by increasing the surface area for them to act. If we talk about the composition of pancreatic juice, it contains a number of lipolytic enzymes. That is, it contains pancreatic lipase, which is used for the digestion of triacylglycerides. It contains phospholipase A2, that is useful for the digestion of phospholipids, and it also contains cholesterol esterase, that is used for the digestion of cholesterol esters. We will study in detail all these reactions in next slides. Secretion of pancreatic juice is stimulated by passage of an acid gastric content, that is acidic chyme, from stomach into the duodenum portion of the small intestine. It is also secreted by the secretion of the gastrointestinal hormones and by the presence of cholesterol esterase. What is bile and bile salts? Bile is produced in the liver and is stored in gallbladder. It is an alkaline solution composed of bile salts. It also contains lecithin, bilirubin. Major function of bile is to give emulsifying agent, bile salts and phosphatidylcholine. What is pancreatic cholipase? It is secreted from pancreas as procholipase and is activated by trypsin. Gastrointestinal hormones. There are 4 gastrointestinal hormones that start the process of digestion and regulate this process. First of all, secretin. Secretin and cholecytokinin are most important of them. However, the 4 are secretin, cholecytokinin, hepatocrinin, and pancreozymin of cholecytokinin. So secretin, it increases the secretion of electrolytes and fluid components of pancreatic juice and act on pancreas to release bicarbonates, ions. Cholecytokinin causes the contraction of the gallbladder and discharges the stored bile into the duodenum. Bile, after synthesis from liver, if not used instantly in the duodenum, it is stored in the gallbladder for some time or for use to be later on. Pancreozymin of cholecytokinin, it stimulates the secretion of the pancreatic enzymes. And the 4th hormone, it is known as hepatocrinin, that is released by intestinal mucosa and stimulates more bile formation, which is relatively poor in bile acid content. Bile salt, how it is synthesized and secreted. Bile salts are called biological detergents. They help in emulsification of lipids by forming micelles. They are synthesized from cholesterol inside liver. They are synthesized and are derivatives of cholesterol. They consist of a sterol ring structure with a side chain to which a molecule of glycine or taurine is covalently attached by an amide linkage. So the chemically we can say that they are sodium glycocholate or sodium taurocholate. Bile salts are required for the proper functioning of the pancreatic lipase enzyme. Bile is synthesized from cholesterol and is converted to bile acid. A primary bile acid is known as, for example, cholic acid. This cholic acid then converts to bile acyl CoA, and in the presence of an amino acid like glycine or taurine, by attaching this amino acid with it, it is converted to bile salt. Bile salts are required for the proper functioning of the pancreatic lipase enzyme. They help in combination of the pancreatic lipase with two molecules of a small protein, which is called colipase. We have already seen colipase at last slide. This combination enhances the lipase activity. Bile salts also help in the emulsification of fats. They are formed from bile acids, such as the primary one is cholic acid. They are synthesized in the liver and stored in the gallbladder for the later use. The primary bile acids, for example, cholic acids, they are found in largest amount. And there is another chenodeoxycholic acid. The primary bile acids enter the bile as glycine-taurine conjugate. After synthesis in liver, it is directly transported to duodenum and then jejunum and other portions of the small intestine. Mechanism of digestion in small intestine. The first and the most important is emulsification. The process of emulsification takes place here. What is emulsification? It is defined as a process of dispersing one liquid in a second immiscible liquid by the help of some electrostatic or hydrophobic or hydrogen bonding interactions between the bioactive compounds and an encapsulating material. Dispersion of lipids into some droplets happens in emulsification. Bile salts, which have a detergent action, they perform this process. It functions to transport cholesterol in the digestive system. This is done by two ways, by the help of peristalsis and with the help of phospholipids also. Then comes another important process, hydrolysis of fat. Hydrolysis of fat, pancreatic lipase is especially important for this process. Hydrolysis of fat occurs in the presence of pancreatic lipase enzyme, and this process occurs at very low speed, at very low rate. We will study this process in detail in next slides. Hydrolysis of dietary phospholipids. Phospholipase A2 enzyme, which is present in intestinal lipase, it hydrolyzes fatty acids at second position of glycerophospholipids and produce lysophospholipids or lysophospholipids. These lysophospholipids being detergents, they help in the process of emulsification and digestion of lipid. We have already seen that in the process of emulsification, we need two processes. Number one, the peristaltic movements, and number two, phospholipid. So this is the lysophospholipid that help in the process of emulsification and ultimately the process of digestion of lipids. Then comes hydrolysis of dietary cholesterol esters. Cholesterol esters, they are hydrolyzed by pancreatic cholesterol esterase to produce cholesterol and free fatty acids. Micelle formation also, it's a very important process that starts the process of absorption. Let's look into detail all these mechanisms. Emulsification. Lipids are hydrophobic, we know this fact already, and thus are poorly soluble in the aqueous environment of the digestive tract. The digestive enzymes, such as lipase, they are water soluble and can only work at the surface of fat globules. That is, they can only digest the surface of the fat globules and not the inner one. So, what is the solution to this problem? Digestion is greatly aided by the emulsification here. The breaking up of fat globules, that is the solution is that breaking up of this fat, large fat globule into much smaller emulsion droplets, such as these one. Triglycerols digestion occur at water interfaces. Rate of triacylglyceride digestion depends on surface area of the interface, which is increased by churning peristaltic movements of the intestine combined with the emulsifying agent of bile salts. The critical process of emulsification takes place in the duodenum. As we can see here that in this diagram that a large fat droplet with the action of bile salt, we have already discussed that bile salt has two sides, hydrophilic side and a hydrophobic side. In the process of emulsification, we can observe that hydrophobic side attaches on the fat globule. As fat globule is also hydrophobic, so it attracts the hydrophobic site of the bile salt. They attach and covers completely one small fat globule, such as the other fat globule, and the all fat globules, they are converted into the small droplets by attaching on the with the hydrophobic sides of the bile salt. While the point to be noted that the hydrophilic side, it remains on the other side where water is present, and help in the process of digestion and help in the process of action of enzymes. Action enzymes, they act on the hydrophilic side of these small droplets. Then comes the process of micelle formation. Micelles, they are disc-shaped clusters of amphipathic lipids placed with their hydrophobic groups on the inside and their hydrophilic groups on the outside of the clusters. Like in this diagram, we can see that a hydrophobic side on the inner side and the hydrophilic on the outer side. Outside of the cluster, there is a cluster known as micelle. Mixed micelles, they are soluble in the aqueous environment of the intestinal lumen. The micelle approach the brush border membrane of the enterocytes. Micelles constantly break down and reform. They remain in the process of breaking down and reforming and reforming. It is the monoglycerides and fatty acids that are free in solution and are absorbed, not the micelles. Micelles, they are not absorbed directly, but the monoacyl glycerides and free fatty acids. Once the process of absorption starts, micelle is broken down into the free fatty acids, and free fatty acids are absorbed across the intestinal lumen wall, the brush border villi. Because of their non-polar nature, monoglycerides and fatty acids can just diffuse across the plasma membrane of the enterocyte. Some absorption may be facilitated by specific transport proteins. Here in this diagram, we can see that there is a hydrophilic polar head, while the hydrophobic non-polar inner hydrocarbon chain. Fat emulsification. Fat globule. The same fat emulsification process is represented in this diagram. A large fat globule, by the action of bile salt and phospholipid, that is by the process of emulsification, and is converted into the small droplets of triacylglycerides. These small droplets of triacylglycerides are then acted upon by the enzyme lipase, the pancreatic lipase here, and are converted into the free fatty acids and monoacylglycerides. These monoacylglycerides, free fatty acids, they are absorbed directly towards the intestinal lumen. But monoacylglycerides are directed to form micelle formation. In short, we can say that micelle formation, they are holding station for some time. A large number of monoglycerides that are produced by the action of lipases from triacylglycerides for some time for the storage purpose, they are converted into the micelle formation. But it is important to note that micelles are not absorbed directly, as they are now a large molecule. So micelles has to be converted again towards the free fatty acids and monoglycerides in order to be in the absorbable form. Then comes the next process, the next mechanism of digestion, that is hydrolysis of lipids. Digestion of fats by the action of lipase is proceeded in steps as, first of all, removal of terminal fatty acid to produce alpha beta diglycerides, that is, triacylglyceride by the action of water is converted to diglyceride. Here a terminal fatty acid is removed and is now known as free fatty acid. The other terminal fatty acid is then removed to produce beta monoglyceride. Another terminal fatty acid will be removed and the diacylglyceride with the help of water will be converted to monoacylglyceride or monoglyceride. Here the second molecule of fatty acid is removed. This monoglyceride, this fatty acid will be absorbed in the intestinal lumen and the monoacylglyceride will be converted to the micelle formation or will be used for micelle formation. Beta glyceride is then first converted to alpha monoglyceride by isomerization of isomerase enzyme and then this alpha monoglyceride will be then hydrolyzed by pancreatic lipase into glycerol and fatty acid. We can see this process here in detail. First of all, from the position 1, this is a triglyceride molecule from the position 1 and the position 3, the two fatty acids and are released as free fatty acids. While pancreatic enzyme, actually pancreatic lipase, it is an enzyme that breaks the bond between glycerol and the fatty acid at position number 1 and 3 more easily. In the next step, this beta glyceride is converted to, first of all, alpha monoglyceride, that is by isomerization with the help of isomerase enzyme. Now the isomerase enzyme shift this fatty acid towards the alpha position, and this is converted into alpha monoglyceride, and then this alpha monoglyceride will be acted upon by the pancreatic lipase and will be converted into the free fatty acid, and ultimately we can get a free glycerol backbone. Alpha and beta monoglycerides, they are just a major end product of fat digestion, and about 25% of ingested fat is completely broken down to glycerol and free and fatty acids. Actually, this last fatty acid, it is linked by secondary ester group, hence cannot be hydrolyzed by pancreatic lipase. Cholesterol esters, with the help of enzyme esterase, they are converted to cholesterol and fatty acids. Here, cholesterol backbone is removed. And phospholipids, with the help of enzyme present in pancreatic lipase known as phospholipases, they are converted into fatty acid and lysophospholipids. These lysophospholipids are important helper in the process of bile salt formation. In all cases, products are more polar than reactants. This phospholipid include both biliary phospholipid that is present in the bile and also the dietary phospholipid. Both of the phospholipids, they are proceeded in the same way. This is the phospholipid degradation, the detailed process of phospholipid degradation. Here the phospholipase A requires bile salt for its activity. The phospholipase A2 enzyme, it removes one fatty acid from carbon number 2. This is the carbon number 2. So first of all, it removes one fatty acid from the carbon number 2 of phospholipid to form lysophospholipid. This is the formula of lysophospholipid. It is important to note here that contradictory to the triacylglycerides, here the carbon number 2 is removed at first step. Fatty acids at carbon number 1 and carbon number 3 are still intact. So this is the formula of lysophospholipids. The remaining fatty acids at position number 1, it can be removed by an enzyme known as lysophospholipase. Glyceryl phosphorylase base that may be excreted in the feces, further degraded or absorbed. Then comes the digestion of cholesterol ester hydrolase. Dietary cholesterol is mainly present in the free form, that is non-esterified form. Only 10 to 15% is present in the esterified form. Cholesterol esters, they are hydrolyzed by pancreatic cholesterol esterase, that is known as cholesterol ester hydrolase, also its second name for this enzyme, to produce cholesterol and free fatty acids. This enzyme, cholesterol ester hydrolase, it is important for the digestion of cholesterol ester hydrolase and is present and is an important part of the important component of the pancreatic lipase. The enzymatic activity is greatly increased in the presence of bile salt. So, the detailed diagram of emulsification and digestion of triacylglycerides. From liver and gallbladder, that is freshly prepared or stored bile salt, when enters through bile duct into the small intestine, from stomach the acidic chyme is entering that has fat droplets, that is along with the carbohydrate and protein diet there are fat droplets that contains triacylglycerides. So these fat droplets, by the action of bile salts, will be converted into, after the process of emulsification, they will be emulsified, they will be converted into emulsified fat droplets and are still now they are triglycerides. These triglycerides were then acted upon by the lipase enzyme and will be converted into the diacylglycerides, then monoacylglycerides, and free fatty acids. Free fatty acids will be could be absorbed directly through the lumen of the small intestine, while the monoacylglycerides would be converted into micelles. Here, some free fatty acids may also be converted into the micelle formation for some time. So the first step is the emulsification of fat droplets by bile salt here. The second step is the hydrolysis of triglycerides in emulsified fat droplets into fatty acid and monoglycerides. And the third step is the dissolving of fatty acids and monoglycerides into micelles to produce mixed micelles for storage purpose. So next is the absorption of lipids. How lipids are absorbed in the general circulation. Next, those products of fat digestion, that is fatty acids, monoglycerides, glycerol, cholesterol, and fat-soluble vitamins. Up till now, we see that these are the end products of the digestion. They need to enter into the circulation so that they can be used by cells around the body. Again, here bile helps with this process. What bile does? Bile salts cluster around the products of fat digestion to form the structures called micelles. These structures, that is the micelle formation, it helps the fats get close enough to the microvilli of intestinal cells so that they can be absorbed. The product of fat digestion diffuse across the membrane of the intestinal cells, and bile salts are recycled back to do more work, that is emulsifying fat and forming micelles. Here glycerol, short and medium chain fatty acids, that is chain length less than 40 carbons. They are directly absorbed from the intestinal lumen towards the portal vein and taken to the liver for further utilization. While long chain fatty acids, that is free cholesterol and beta acylglycerol together with bile salts, they form mixed micelles. This diagram shows the process of absorption from intestinal lumen. Large lipid droplet, by mixing with bile salt, they are converted to micelles. And micelles, by the action of pancreatic lipase in the intestine, they are converted to free fatty acids and monoglycerides. And then these free fatty acids and monoglycerides are transported towards the epithelial cells of the mucosal cells of the intestinal wall. We can say here that the free fatty acids and the monoglycerides, they enter in the intestinal wall, the mucosal cells, and here are then again converted to these monoglycerides. They are then again converted to triacylglycerides. That is the triacylglyceride synthesis occur here at the endoplasmic reticulum, and also the formation of chylomicrons. What are these chylomicrons? Chylomicrons, they are a combination of different end products of fat absorption or fat digestion here. For example, it contains monoacylglycerides, triacylglycerides, fatty acids, cholesterol backbone, glycerol backbone. These chylomicrons are then transported towards the lymphatic vessel and towards the specially muscles such as cardiac muscles or other tissues of the body. The mixed micelle that contain, for example, monoacylglycerols, it also contains long chain fatty acids, it contains cholesterol, it also contains short chain fatty acids and the glycerol that are end products of triacylglycerols, and is surrounded by the bile salts. These bile salts remain here and are transported towards the hepatic circulation. We will see hepatic circulation in next slide. While here all the contents of the micelles, they are transported towards the intestinal mucosal cell, where monoacylglycerols, for example, two monoacylglycerol, they are converted to ultimately triacylglycerols here. Free fatty acids, by the action of enzyme thiokinase, they are converted to fatty acyl-CoA, and cholesterol is again converted to cholesterol ester. Within the intestinal epithelium, monoacylglycerols are hydrolyzed to fatty acids and glycerol, and two monoacylglycerols are recycled to form triacylglycerols via the monoacylglycerol pathway. Lysophospholipid here, They are recycled to form phospholipids also, while cholesterol is acylated to form cholesterol esters. Long-chain fatty acids are used for esterification to form triacylglycerides, phospholipids, and cholesterol esters. Short and medium-chain fatty acids are released into the portal circulation and are carried by serum albumin towards the liver. This is the summary of the whole digestive process that we have already seen yet. Now, after the resynthesis of triacylglycerols and cholesterol esters, the bile enters in the enterohepatic circulation. That is, what is the enterohepatic circulation of bile salt? The bile salts, they are present in the body but are not sufficient to fully process the fats in a typical meal. Thus, they need to be recycled and must be used again and again. This is achieved by the enterohepatic circulation. Specific transporters in the terminal ileum move bile salt from lumen of the digestive tract towards the intestinal capillaries. From ileum, they move the bile salts from lumen of the digestive tract towards the capillary bed, intestinal capillaries. They are then transported directly to the liver via the hepatic portal vein. This is the hepatic portal vein. Hepatocytes take up bile salt from the blood and increase the secretion of bile salt into the common hepatic duct and common bile duct and enters again in the duodenum. 95% of the bile that is released to the small intestine is then recycled again via this process, via the enterohepatic circulation, while only 5% bile salt is excreted in the feces. This diagram also shows the formation of chylomicrons, that is triacylglycerides and bile salts along with lipase enzyme. They are converted to fatty acids and form mixed micelles along with monoglycerides. These fatty acids and monoglycerides, they enter the brush border villi and inside the mucosal intestinal mucosal cells, they are converted to again monoglycerides and fatty acids again converted to triacylglycerides and are transported and made chylomicrons. These chylomicrons are then transported towards the circulation. This is the diagram of chylomicron. We can see here the structure of chylomicron, that it contains particles for transport of lipids to liver and adipocytes. Its size is around about 1 micrometer and average composition is that here triglycerides are 84%, cholesterol is 2% and some other percentage, that is 4%, 8% and 2% other complex fats. There are some apolipoproteins on its surface. How transport and utilization of chylomicrons is brought about. From intestinal tract, these chylomicrons are transported towards thoracic duct. From thoracic duct, it enters into the venous circulation and passing through systematic arteries, they are transported towards the peripheral capillaries. The fate of these chylomicrons is that they might be converted into free fatty acids and monoacylglycerides and are transported towards liver, or they may be directly transported towards skeletal muscles and cardiac muscles, or might be stored in the form of adipose cells. What is the fate of dietary lipid? Triacylglycerides, they are converted to free fatty acids and glycerols. Free fatty acids, they form from triacylglycerides, they are transported towards muscles for energy production or adipocytes, and glycerol enters from triacylglycerides, it enters in the liver and forms glycerol 3 phosphate and is used in glycolysis or gluconeogenesis, that is the process to make a glucose molecule. While the chylomicron remnants, they are endocytosed into liver and are hydrolyzed to their component parts and recycled by the body. That is, it might be converted into free fatty acids and monoacylglycerides, or might be transported towards the skeletal muscles. If this process is decreased due to impaired binding to the receptor in liver, they accumulate in the plasma, leading to type 3 hyperlipoproteinemia. This is the hormonal control, how hormonal balance regulation occur. In stomach, gastric motility cause the breakdown and the chyme The acidic chyme enters the small intestine. Here, in the small intestine, dietary lipids are broken. Here, degradation of dietary lipids occur. While from the gut cells, that is the endocrine cells of the gut, they release cholecystokinin and secretin, two hormones. That is the increased production of and the digestion of dietary lipids in small intestine stimulate the gut endocrine cells and release two hormones, cholecystokinin and secretin. This cholecystokinin decrease the gastric mobility, and hence this process is might be stopped in order to increase the satiety value. Or it may release secretin. The secretin act on the pancreas to release bicarbonate ions, pancreatic lipase. Here is the physiologically important lipases such as the lingual or acidic stable lipase that are present in mouth and stomach, and act on triacylglycerides with medium chain fatty acids, and this is the product that is made, the free fatty acid and diacylglyceride. Similarly, after pancreatic lipase, intestinal lipase, and phospholipase A2 that is present in small intestine and act on phospholipids. Lipoproteins, lipase, insulin that is present in capillary walls and act on triacylglycerides in chylomicron or VLDL, and triacylglycerides stored in adipose cells and convert them into free fatty acids and glycerol backbone. The abnormalities of mild digestion due to maldigestion or malabsorption. These are the tests for assessing fat absorption and malabsorption and test for pancreatic function. The summary of the lipid digestion and absorption, starting from oil droplets or oil drop by the action of lipase or colipase and lipase enzyme and colipase protein. This oil droplet is converted to triacylglycerol, monoacylglycerol, fatty acid. That is the emulsion droplet. This emulsion droplet then by the action of bile salt is converted to mixed micelle. This mixed micelle is then absorbed through the enterocytes towards the mono and is then again used for resynthesis of triacylglycerides, and chylomicrons are then transported towards the general blood circulation. From here bile salts are recycled. Thank you.